So Danielle asked me to replace her, and I'm speaking, but she did all the work that you will see today, and she's here to answer questions also. For several years, we are interested in the involvement of the folic acid cycle and mainly the contribution of the methylene tetrahydrofolate in neurodevelopmental disorders. And this illustration just to point out to the involvement of the products of the cycle in other very critical processes like DNA synthesis needed for cell proliferation and methylation, regulation by methylation, and the other processes that I mentioned here. And about 10, even a little bit more years ago, first epidemiological evidences for the polymorphism in a place where encode for the catalytic subunit, an amino acid in the catalytic subunit of the enzyme, increasing as a risk factor for autism, and it was shown previously also as a risk factor for schizophrenia and neural tube defects. And interestingly, or maybe not like surprising, that the polymorphism in homozygotic form in the mother is also increasing the risk. So it's kind of an example of interaction between the in utero environment and the genetic of the offspring itself. So in a mouse model that we first characterized in the adult age, we managed to show in 3 independent studies that the heterozygosity in the mouse, which actually produce 50% of the activity, more or less resembling the homozygote form in the human, cause autistic-like features, behaviors, and it was also associated with changes in the inhibitory system. And in recent years, Noah managed to show developmental delay in these pups of the heterozygote mother and deficient ultrasonic vocalization, both in the usage and the properties of the call. So considering all these known evidences, we focus now on the early stages, whether we see these changes in the inhibitory system at early stages. So just to remind you that all interneurons in the brain, and now we focus on the cortex, are generated in the ganglionic eminence, and from there they migrate to the rest of the brain and to populate the cortex in very spatial pathway that first they navigate toward the cortex, and then they penetrate the cortex and populate the layers. And we all in this room know that the location within the layers is very critical for the circuit. And this is happening from the second trimester to the third trimester of pregnancy or embryonic period. A little bit later, and early postnatal in human and mouse, 2 transporters that control chloride, which is the ion that move in the inhibitory synapses, changing their expression when the NKCC1 in pink here, the expression is going down with development, and the KCC2 expression increase. So altogether, that's formed this shift in the concentration of chloride from high level at the embryonic period to low level in postnatal life, and this is also affecting the inhibition in the GABAergic system. So Danielle first in the control mice just checked the expression of these transporters in the brains, in the cortices of our mice, and some GABA subunits. And we see here that some of them have a steep increase, and some of them moderate, and some of them didn't change. So more or less, we show, or she managed to show, that the time course of these events in our mice cortices. And the hypothesis for her experiment was that the deficiency in the GABAergic system begin early in development, and that's what she set to check here. So she showed previously some evidences for changes in proteins, and now we move on to new stuff. So the experimental design is the control group, the wild-type mother, ++ for the MTHFR, and heterozygote mother that gave birth to either heterozygote offspring or a wild-type offspring. In all this experiment, in black is the male, because it's C57 black, and it's expressed TD tomato in all interneurons, so we have some marker for the interneurons in their brains. And first, I will show you this interesting data, and then we jump into this TD tomato. So what Danielle did, she picked 3 genes from 3 that represent chloride transporters, the 2 that I showed you before, and one of them have 2 isoforms. 3 genes representing the BDNF signaling, and 3 genes encoding for GABA A receptor subunits, different. And she checked the expression, and in addition to the traditional analysis, what we show here is the correlation in the level of expression between these 9 genes. So it's not so easy to see, but in general, the transporters are below the titles and the same over there. So this is the control group in your left side, and we see the difference in the pattern of expression. Some of them are highly correlated, indicated in red, and some are highly discorrelated, negative correlation shown in blue. And we see that the pattern is different between postnatal day 7 and 0, which is normal like developmental course. So this is what we compare to. And here are the other groups, the wild type offspring to the heterozygote mother and the hetero offspring. And first we took a look at it, and we say, okay, it's very, very early stage at postnatal day 0, and we already see this interaction between maternal genotype and the offspring genotype, when the maternal genotype alone was sufficient to show a very different pattern compared to the control. And the second thing that attracted our eyes is the similarity between the wild-type offspring here in the second layer here, which is, it looks very similar in some ways to the postnatal day 0 control. So it's still something that we're looking at, this combination of genes and the genes that are coordinated in their expression. And we don't really know exactly how to interpret it, but it's given us a direction about the link between these groups of genes. Something else that Danielle checked is just to be sure if we see these changes in the inhibitory system, maybe like Evan before mentioned, this excitation inhibition, maybe something else also is going on in the glutamatergic receptors, which also change the proportion around the similar age, and in a way they're more or less following the same pattern at postnatal day 7. So this is just something to tease our thinking about it, and now we'll take you further to something that looks to us even more like bread for the brain for thinking. So we spoke about, I showed you before, this path that the interneurons follow into the brain, and to capture this complicated navigation of the interneurons, Danielle took 3 samples. She fixed the brains of postnatal day 7 mice, and she sliced them and made thin sections, and it is illustrated here, the positions where she sampled rostral, medial, and caudal sections as shown below. And in each of these sections, section, she took pictures representing the midline or close to the midline in the cortex and lateral position. So we have kind of not exactly 3D, but somehow 3D of the brain. And this is an example of how it looks with the interneurons locations where she sampled. And to capture the migration through the layers, she divided the brain from the pia to the corpus callosum to 10 equal beams and analyzed them separately. And later on, she grouped them based on this functional or regional division of layer 1, cortical plate or cortex, intermediate zone, and subventricular zone, ventricular zone for the analysis. So first she looked at the number and density and position of interneurons, shown here in red. And then she looked at the NKCC1, one of the chloride transporters that I mentioned before. And if we know like the developmental path, we expect some changes between the rostral and caudal regions and also between the layers. So it was very important for her to capture the whole picture. These are the results in these 3 groups that I mentioned before: rostral to caudal and lateral to medial, and in blue is the control group and then the 2 offsprings. Wild type in orange and heterozygote in gray. And you can appreciate first of all, the normal distribution in the blue lines between layer 1 and the rest of the layers. And there are some natural changes between the rostral and caudal areas. But for us, it was very interesting to find out that the heterozygote offspring in layer 1, both in medial and lateral positions, show significant increase in the number of interneurons, which normally are expected to be very low in this layer. And then we see that in different areas of the brain, in the anterior to posterior axis, the effects are very different. So if we take the whole brain and homogenize it and then measure something, we miss this picture, which is very important and showing some critical differences in interneuron distribution. And this is the picture for the NKCC1 expression, density of immunofluorescent. And here we see that the main effect in the heterozygote offspring are in the caudal regions. And in general, if we look at the blue bars, we see that from rostral to caudal, we expect some natural differences, like the expression of the molecule is not similar in the same layer at all the brain. So these differences, first of all, we try to, again, make sense of them. What does it say? And one thing that we compare to is this scheme of at what age we expect to see what. So embryonic age 18, we expect that these streams of interneuron migrating to the cortex will include relatively many neurons, and then with the age until postnatal day 6, they are all concentrated in the cortical plate, and this is not what we see in the experimental groups, and this is kind of summarizing the differences that we see. This significant effect at rostral regions in layer 1 and other layers, and also in the distribution at our other brain regions, but it somehow hints that the brain that we see at postnatal day 6 of the heterozygote offspring is not exactly showing the characteristic of the organization of interneurons at the matching age in the control, not the expected one. And this is the NKCC1 expression. We're still waiting eagerly to know what happened with KCC2 that Daniel just began to analyze. So altogether, to summarize, cortical genesis and interneuron and also excitatory neurons follow some developmental path which is very nicely three-dimensional organized the temporal and the spatial orientations, and we see that this is not what's happening, or that there is dysregulation in this organization in the heterozygote mice. But more than this, which is very interesting for us and we don't have any answer, is how much the interaction between the wild type, the heterozygosity of the in utero environment and the genotype of the offspring, how come they show so big diversity? And in some ways, the heterozygotes show very strong phenotypes, and in adulthood, the wild-type offspring show very strong phenotype. So it's still a lot of work to do. But we think that this is part of what's causing the heterogeneity of the phenotype, this interaction between the in utero environment and the genetics of the offspring. So Danielle did all the work that is here, inspired and supported by the following here, Noam and Ayala, that work in related projects. And the future of this year, part of the students sitting here, will try to correlate behavior to the finding in the brain by performing the same experiments, the behavioral and looking into the brain in the same mice. And in the far side, the past students that contributed to the building the knowledge. Thank you.